Hello, my name is Bryce McCourt. This is my presentation on sulfuric acid and its current applications. This current application being discussed today is specifically within the realm of environmental monitoring. This is for the Concepts of Analytical Chemistry class at Grand Canyon University on the online format. So first, in regards to a brief overview and description of sulfuric acid, this chemical is actually recognized as the world's largest bulk chemical with an annual production of 201.6 million tons. It is utilized in the production of things such as fertilizers, dyes, pigments, detergents, drugs, inorganic salts, and acids. It is used in petroleum refining and iron and steel production. It also aids in the production or the extraction of elements such as mercury and scandium. And specifically for mercury, what happens is sulfuric acid is actually a chemical that is added to a precipitation reaction that is then brought about. And what happens at the end, once the right temperature is then there, is that mercury crystals are then precipitated out for the reaction. Sulfuric acid is also corrosive with metals and tissues, and it has been known to char wood and other organic matter on contact. And it can be a concentrated acid that oxidizes, dehydrates, or sulfonates most organic compounds. And there are specific techniques that can be utilized for the measurement of sulfuric acid. And the first one is the barium-thorium titration method. And in this way, a gas sample is first extracted from a stack, and in this particular case, sulfuric acid was wanting to be separated from sulfur dioxide fumes, so they were using this titration method. And first, a gas sample was first extracted from a stack, and then a thorium indicator was added, and the sample was then titrated with the barium standard. Another way that it's measured is through ion chromatography, and this specific technique is used for the determination of sulfuric acid in ambient air. So regarding its current application, and this is specifically in environmental monitoring, the problem is regarding hazardous waste material that comes from hazardous waste treatment. And so what happens in the European Union specifically is for their hazardous waste, they were incinerating it. But then they found out that incineration was not going to be the absolute solution to their problem, because what was happening was residuals were remaining after the incineration. A very high percentage, up to 25% of what was being incinerated was being left as residues. And in these residues, there was high concentration of metals, such as iron, chromium, manganese, nickel, copper, zinc, lead, tin, and antimony. These specific metals were remaining in the residuals after even being incinerated, and then these could then be classified as hazardous waste materials. And so to combat this problem, there was a need for sulfur oxidizing bacteria, and this is a process known as bioleaching. This process can also be dealing with iron, but since we're talking about sulfuric acid, we're going to specifically mention the sulfur oxidizing bacteria. Again, the process is known as bioleaching, and it's in the treatment of these residuals with the potential for recovering those valuable metals that were left behind in the residues and also reducing the toxicity that is left behind because of those metals. And regarding the analytical method that is used, what they first do is that the sample was centrifuged, and what they want to do there is they want to remove elemental sulfur and the slag particles, because then they want to determine the number of cells, so the cell number determination. And they also want to go about getting optical density measurements, specifically at 660 nanometers. They want to do that using a spectrophotometer. And also, they want to find that right pH there for the sulfuric acid. So pH measurements need to be taken using a pH meter with a glass combined electrode. And also, there needed to be a process to go about. This process was needed to identify optimal growth conditions and the maximum rate of sulfuric acid generation for the bacteria. So pure cultures of both sulfur oxidizing bacteria were tested at different concentrations of sulfuric acid, and then they were also tested at different temperatures. And as they went about this, they took samples of pH, they took samples of optical density, and also because we're talking about sulfuric acid, they also want to go about getting sulfate measurements that were collected every 2 to 3 days. And we not only want to go about just with original sulfuric acid, they were wanting to test biological or biogenic sulfuric acid because we're talking about bacteria. So biological sulfuric acid production, it was tested in a continuous stirred tank reactor versus just normal sulfuric acid. And the analytical method for the total acidity measured, it showed only minor standard deviations during the entire batch experiments. And we want to go about that with analytical chemistry because we want to focus on standard deviations and errors and keep them rather small. And what they found in their main study, the main findings of the study, was that the sulfuric acid produced to control the pH in regards to the bioleaching efficiency percentage, it was the highest at approximately a pH of 2.0, specifically with the biogenic sulfuric acid. Again, they were wanting to specifically look at normal sulfuric acid versus biogenic sulfuric acid because we were wanting to work with sulfur oxidizing bacteria. Next, the bioleaching efficiency percentage for the biogenic sulfuric acid, it was higher just compared to that normal original sulfuric acid. Next, the bioleaching experiment resulted in a relative abundance of both iron and sulfur oxidizing bacteria, along with other bacteria that was present as well. Again, that bacteria it needed to grow specifically in those acidic conditions, and we were testing it. And finally, the bioleaching efficiency percentages for each metal, they were the highest for specifically the biogenic sulfuric acid. For the sulfuric acid was needed to control the pH to result in the high bioleaching efficiency. Again, we needed that sulfuric acid to stabilize our pH for that bacteria. And just to give a visual representation of what the results of the study were, this is an example of the sulfuric acid produced to control the pH in regards to the bioleaching efficiency. It was the highest at approximately of the pH of 2.0. And again, this is specifically with that biogenic sulfuric acid. So if you would look, there's a bunch of data here specifically where it's at the 2.0 for the pH. The x-axis was representing the time that was in days, and the y-axis was representing the total acidity, specifically in molar concentration. Another result from the study, this is specifically the bacteria, that iron and sulfur oxidizing bacteria results. And the bioleaching experiment resulted in a relative abundance of the iron and sulfur oxidizing bacteria, along with other bacteria that is present. So the top one was specifically at 30 degrees Celsius, and they have different percentages of sulfur that was present in them. And then B represented the bacteria that was growing at 37 degrees Celsius, again at the different percentages of sulfur that was there. So again, they found that there was both an abundance of the iron and sulfur oxidizing bacteria. All right, this graph for results of the study, this is showing the bioleaching efficiency percentages for each of the metals that we were wanting to recycle and reduce the toxicity in those residues. And the bioleaching efficiency percentages for each of the metals, they were the highest for the biogenic sulfuric acid. For the sulfuric acid was needed, again, we want to control that pH to result in the highest bioleaching efficiency we can get. So the green was representing the pH of 2.0, and it was specifically the biogenic sulfuric acid. And as you can see, the green bars for each of the metals, they are the highest. The white bars, they are for the pH of 2.4 that is biogenic, but they are not as high as the green. So that was a little bit more not as acidic. The 2.4 pH was not as acidic. And then the orange represented the pH of 2.4, but that was with normal, original sulfuric acid. That wasn't specifically classified as biogenic sulfuric acid. And then the blue and the orange circles there, they're specifically telling us the bacteria that was growing in specific pHs. So, for example, the one represented for B is the pH of 2.4, and C, that is specifically the one for the pH of 2.0. And we see that there is a relative abundance of the bacteria. And this specific result, this is just some numbers that I found from the article talking about the incineration of the waste, but how we're wanting to extract those metals and reduce the toxicity and recycle them and really save them to where they're not classified as hazardous waste materials. This is just specifically each element and what the bioleaching efficiency percentage was, and that's going to be in this section here. And then these sections tell us the potential recovery and the amount that was actually recovered. And they are higher whenever the bioleaching efficiency goes up for the metals. So regarding the benefits towards societal benefit for this, first, the use of sulfur oxidizing bacteria, and what we did here, it oxidized zero valent sulfur. We wanted to oxidize it to sulfuric acid for the bioleaching efficiency. This plays a role by making the heap bioleaching an attractive processing technique for this. Next, factors such as low process costs, operation under environmental friendly conditions, and the suitability for processing low-grade materials, this plays a role in the bioleaching using sulfur oxidizing bacteria. And finally, bioleaching, this supports the idea of comprehensive utilization of resources. So we want to use as much as we can and utilize as much as we can. And specifically in this example, it was the metals that we recovered and recycled. And when we recycled them from the residues, it makes them economically attractive, and it doesn't classify them anymore as the hazardous waste material because of the bacteria that we have that we got from the sulfur that was then oxidized to sulfuric acid. So in regards to the instructional component as to how I would teach this in a classroom, I have used graphic organizers before in my science classrooms. I have used them at the high school level when I student taught at the high school level. I have used them in a 5th grade elementary class when I taught there last year, and I have also and still use them for my 7th grade science classrooms that I am teaching now. So I have used them in elementary, middle school, and the high school setting. And there is evidence behind teaching chemistry topics using graphic organizers. And facilitating learning a course, as difficult as chemistry can be, it can be made through integrating graphic organizers into the curriculum. And graphic organizer-based presentations, they have indicated a positive attitude from the students whenever they are brought about in a classroom in a study. And incorporating different graphic organizers in identified formats such as a web diagram, a flow chart that's specifically a concept map, a Venn diagram that might be comparing different things and writing how they're the same there in the middle where the circle is, and just picture graphics, they have been seen and obtained as very good perceptions from the students. And I believe from my experience in the classroom at all 3 levels, I think it helps students organize different concepts and different materials, especially material and concepts and skills that they're learning that might be a little bit more on the difficult side, which is chemistry can be a little bit on the difficult side. So to help students maybe organize their thoughts and shape them out, I think graphic organizers really do the trick with that, really at any age. And so this could be a specific graphic organizer example I believe I would use. And so you would have a chemical name here at the top, and then you would have sulfuric acid here for our example. And then the students would then go through, this could be the description part, and then here they could write out the description part. And then another one that they could go about doing is the connection to the environmental science. So the environmental monitoring that we went through, or they can even research another example and write it in the blank. They could then talk about the important uses of sulfuric acid. They could also talk about the techniques that are utilized for the measurement of it. They could then go about and research the connection to it with biology and biochemistry, like we did with the bacteria just now, and that was kind of even connecting environmental science to it. They could also discuss and research toxicological effects, and then also any unique characteristics of sulfuric acid. And again, these are kind of in columns, and it will help them organize their thoughts. And I think that will really help them organize important information, not just really jumble it up. So I believe if I was teaching a material such as this, I would try to implement some sort of table, graphic organizer, even a concept map that might say sulfuric acid in the middle, and then extend out with different ideas. I believe I would go that route. Okay, these are my references that went along with my presentation. Thank you very much for listening.